see how good it performs against the null situation. And this allows us to control the covariance. Figure you're calculating hundreds of test statistics. You don't want to do something like Bonferroni in some of these situations where things were highly correlated, and you want to control the overall error rate. And so this provides it's just basically a mechanism to control the error rate. And so I don't want to go through all of a simulation study here, so I'll get to the guts of it just in terms of a curve here. If you just tested in the situation, I'll go into too much detail. This is the size of the interaction, so basically how much that color intensity changed over the way. And then if you just did a marginal test statistic, this black line, that might be the power. Because even though the interaction is increasing, that's inferring some marginal effect in this situation. You could estimate all of those parameters, those alphas before, which basically I'm going to fit this big whopping model with maybe 5 or 10 parameters and see if I can find that interaction. That actually does better, at least it wouldn't always do better, but in this situation, which is fairly well controlled, it did better than the marginal test. And then this says, okay, I'm doing variance control. The stage-wise method, or basically that estimation procedure and weighting, and you get better power there. And so think of this as another filtering device where I may have only picked out 1 or 2 variables by filtering on these interactions that I can use later on and find out what's going on. But variance control is critical. We always design these studies for a main effect. We barely power them up to answer the treatment effect. So even though those associations may be real, we have to be realistic in the fact that there's a lot of variance in our systems. At least in oncology, there tends to be a lot of noise when we're looking with patient outcome data. And this was an example of actually trying it in a myeloma patient population. Our endpoint here actually was, and it was SNP data, our endpoint was a combination of early death and progression, sort of like how bad does the patient do immediately. I'm sorry, my PowerPoint, I put that slide over top of my text there. But we calculated these test statistics, and the coloring is like I had before, where yellow was the marginal, green was doing a little bit of adjustment thing. So you can kind of think of this is how much I shoved those test statistics larger. Of course, that's not entirely fair. I have to pay for all that looking. So down here, I've sort of said this marginal test statistic was 3.29. It increased doing this stage-wise weighting went up to 4.76. But if you try to calibrate for all that looking in some, sensible way, that the family-wise controlled p-value is still 0.031. So we found something. We probably wouldn't have found it doing the multiple comparisons on the marginal test, but we did find it by doing the weighting in this example. So if there are interactions, and I believe there probably are, you can help yourself a little bit. Uniformly, not necessarily. So on the West Coast, we don't think about Florida when we're thinking of sunny weather. We always think about Hawaii, so I had to have my schematic of what I'm trying to say that's more generally model-based. So we're trying to think of, really all of these methods are think about gating. We're trying to gate the therapeutic effect or gate the environmental effect. And the analogy I came up with, and I'm told this isn't particularly helpful by my colleagues, but I still love it anyway, so I'm going to try it yet again. So if you're looking at the Pacific Ocean, the cool thing about Hawaii is not necessarily the palm trees. We know there's other places with palm trees. The cool thing about it is the fact that there's a weakness in the crust, and those islands have formed as the openings have moved over top. It's created these volcanic islands, and if you looked at the ocean in general, you wouldn't have found it. But if you could focus in where the weaknesses are happening, you see where that stuff is coming through. So that's my analogy. If you can construct algorithms in a fairly principled way, focus in where there's some therapeutic activity, then you should be able to find it. And I should say, following off, it's again good to work with good students, and Nino Pasheva at the University of Washington is working on something called directed boosting to try to come up with these in a reasonable way. How are we doing on time? Okay, so this is the last component of ad hoc ideas to try to filter through data, and this is one that I've been thinking about recently because in my collaborations, there is so much pressure to make quick decisions. So can we use those interaction tests to really help us design studies on the fly or at phase 2? And so this is sort of the attempt to do that and figure out what the properties are there. So first, it's good to remind ourselves what phase 2 trials are good for. They're good to decide what you're going to do for phase 3 studies. everybody wants them to look at. I should also, since I'm on being videotaped, I should also mention the fact that this slide was really partially stolen from Rich Simon at the NCI, so I've got my disclaimers there. You want to, and then nowadays you want to identify biomarkers of interest and be able to screen for subpopulations. The question is, how do you have to formally test this in phase 3, but the thing you're formally tested might be based on a relatively small sample size. You could commit yourself to the wrong direction early on. So what implications does this have? Clearly you want to do it, but again, you want to be careful. And I get pushed to do this a little bit, and I think it's a good idea, but it's good to think about it. So the basic idea is looking at the data as a variable process. So these subgroups that I'm going to design my phase 3 trial on, say individuals with gene expression above 25.3, might be too aggressive of a look. So how should it be able to be used? And this is, again, repeating that we just get pressure to do this all the time, at least in my opinion, at many levels, because people, you want to commit resources, you don't want to spend too much patience, you want to sound innovative. There's a whole section in your grant application called innovative. You want to be innovative, but what's the impact of being too innovative? So here's a design of a proposed Southwest Oncology Group study. Again, reality always seems to look somewhat more complicated than the statistics getting there. But basically, what this happens, without going into too much of the details, is there's patients that are accrued. You test some markers, you do some modeling. If you're so lucky to find some things that look like they're predictive of treatment efficacy, you use them to develop a population you're going to test in your new study. The good thing about it is, if you don't find any, then you're just going to drop the biomarker analysis. So if you do some testing up front to say, did I really find something or not? So do something to assess reality or not. Of course, what this sometimes turns into is, I do this estimation, I find my subgroup, I dump the other ones, and that might be the problem, because you're not really, you're basing this on a small sample size. I thought I was going to have a bit more time and go through all the components here, but the basic guts is learn on the fly, make a decision, guide your analysis based on what you've learned on the fly. But if you're smart, how you choose this analysis, you probably want to cover your bases at the end and maybe test what you would have tested anyway without the adaptation. That's the saver in it. So I did a simulated example, but not to go through too much of the details. Generate some data, some prognostic factors, some efficacy modifiers, interaction variables, and a treatment indicator. I'm assuming I'm in the phase 2 setting with some historical data, but think about the model you have here from before, which is there's some prognostic, there's some variables that are prognostic, there's also effect modifiers in V as well. I don't list the main effect there. And then we generate some data, and this is a little bit more complicated. I had some historical data and some phase 2 data, I mixed them together. But I'm doing an analysis that's based on these subgroups. So the marker value is saying, I'm going to look at those regions R that I described many slides back and said, I'm going to do an analysis on each one of those analyses, and I'm going to guide, and I'll find the best one, and I'm going to do my primary analysis on that subgroup. So basically, I generate some interaction data. I'm going to guide my decisions based on this interaction. I'm going to do the simplest thing possible. I'm not going to do complicated modeling at all. I'm just going to look for a simple subgroup on a single variable. This is pretty straightforward. What does that mean in terms of power? There's lots of test statistics listed here. I'll just go into a couple. But this is saying there's a main effect. Here, I'm going to say there's an interaction effect. So this represents 2 treatments, and this represents 2 treatments. It was the underlying simulation model. Here is the main effect term. Forget about all this adaptation. I'm just going to run a dumb clinical trial. I'm just going to look at the main treatment effect. This is one, this was a complicated one of weighting between the 2 of them. This one said, this one down here, this pink one, said, I choose the best subgroup and I'll choose the overall test. Kind of like, I'm not sure. And I'm going to choose both of those together, sort of trade off my alpha, control my type 1 here. And if you look here in the main effect, I didn't lose hardly any power by doing this very simple combination of doing the adaptive thing, but also saying, since I don't trust the adaptive thing, I'm going to blow some of my type 1 error in the other one. The results are pretty much the same in this small sample size. Doing this one and this one. This one, select is two versions of these, choosing the best one and following that alone. And you see you lose a fair amount of power just because sometimes you were tricked in thinking it was a subgroup, and really the treatment worked overall. So it's a usual statement, that you went between those two. If there is an interaction, that's where you buy a lot, because if you'd just done the main effect, you'd be down here under this black line. But using any one of these weighted ones, main subgroup combinations, you retain much of that power of the interaction. And even select, which should be the best as far as an interaction, I'm just going to pick that group that works the best. You'd think it'd probably be the best strategy. It still loses because, you know, if the